Namaste students. Today I am going to discuss about the topic single domain antibody. What is the single domain antibody? In the previous video we discussed about the single chain antibody and where we were using variable fragment of light chain in the heavy chain. Okay, variable fragment of light chain and heavy chain. That was the single chain antibody. But in the single domain antibody, we are using that are also known as the nanobody. We are using a DNA fragment of only single monomeric domain. That domain may be either a light chain or heavy chain, not the mixture of both the heavy and light chain. Only either light chain or either heavy chain will be used for the construction of a complete antibody. Okay, it had a molecular size of 12 to 15 kilodalton and mostly very smaller than the common antibodies and comprised of only one heavy protein chain or any light chain, not the all these two fragments. So it is very smaller than the original antibody. So it is the monomeric and specific for a particular kind of specific antigen, specifically designed. Okay, it is these are specifically designed for a specific antigen, not for the any kind of. Okay, this is not a general antibody. It is for the specific kind of antigen. And one more important thing is that these antibodies are used in the laboratory conditions. So that's why they are designed here. This is the diagram of variable part of the heavy chain of one mm, DNA molecule, one antibody molecule. Here only the heavy chain parts of the variable region is used. Okay, what happens for the single domain antibody? First, single domain antibodies were engineered were first from heavy chain antibodies from the camelids, and these antibodies which are isolated from the heavy chain antibodies are known as the VHH fragments. Okay, uh, on the basis of the origin of the organism, they can be named differently. Okay, so there are different methods for the construction of these single domain antibodies. So how we construct these single domain antibodies? First, we need to digest the antibody. Then, after digesting the antibodies, uh, we take only the variable fragment of heavy chain. Okay, we are taking the variable fragment of heavy chain. That heavy chain part will be engineered for the construction of the complete single domain antibodies. So that is the main basic principle. What is the properties of single domain antibody? It is a single domain antibody which have a peptide chain of 110 amino acids, and these peptides have similar affinity to antigens as whole antibodies. Means we do not need the complete antibody of higher molecular weight. Only small 50 kDa, 15 kDa is enough for the identification of the antigen. So that is the major advantage of this single domain antibody. These domain antibodies can be heat resistant and can be spoiled by the detergents, and high concentration of urea, so it must be avoided. So now how these can be produced? uh, these heavy domain antibodies are produced from the isolating the heavy chain. First, the heavy chain of antibody will be isolated from any kind of antibody, and this heavy chain part will be used for the construction of a complete antibody. So this part can be added to another antibody such as IgNAR for the faces, and this is the HCl-GG which can be used. Then another method is that we can construct these single domain antibodies from the common IgG. That is the IgG, this is the IgG, and four chains of these IgGs can be used for the construction of single domain antibody. What are the potential applications of single domain antibodies? Mm, these single domain antibodies are constructed for the purpose of the laboratory purposes mostly, and for the these are used for the biotechnological and the diagnostic purposes. Uh, suppose that one microorganism is very severe, one microorganism is very uncommon and cannot be identified by the general uh, procedures. But using these domain antibodies, we can prepare them by only a variable region of the heavy chain. From these variable region, we can just design our antibodies and then can use these antibodies for the identification of the particular kind of specific antigen for the diagnosis of the disease in the human. So most of them, their work is for the diagnostic purposes for specific disease. Okay. The next thing is that they can be used for the therapeutic purposes also. For example, in the cancer cells. Okay, these can specific single domain antibodies can be used for the binding to particular cancer cells. We can design some single domain antibodies to bind to antigens like HER2 for the treatment of the cancer, because these uh, single domain antibody will bind to these cancer cells, and after binding to these cancer cells they can be used for the killing of the viruses as well as for the removal of the cancerous cells. So these are the purposes which by which we can use these single domain antibodies. Here are some examples which are which are given for the single domain antibodies. So our lecture is over here. Thank you very much.